Okay, good afternoon, everyone.
Good afternoon, Asha.
Um, Li Tong, I don't know, no students stay Huangshi.
没有老师。
哎呀 I hope my students maybe quickly come back Huangshi. And now this space disease, the COVID-19, make all of the peoples having heaving burden. And this semester, the medical school arranged me teach all of you medical molecular biology. Last semester, we studied the biochemistry. All of students maybe grasp many things. You can see the biochemistry studies the chemical reaction in human being's body. And this part, and several years ago, the medical molecular biology, you can see this part, you can see on molecular level, studies the reactions, studies the mechanisms, it is so on. So this part, several years ago, ascribed to the biochemistry. And from the 1953, the Crick and the meeting and founded the structure of the DNA, the double helix structure. And from these times begin this part, the doing many research focused on the molecular level. This part developed so faster. So this part you can see and become the solely subject, the molecular biology. Okay. Uh, this semester we started this part. I want to teach all of you the several chapter. The first we study the introduction and the gene. What is the gene? Maybe normally we meet all kinds of the films refer to the gene. What is the gene? In fact, it is very easy. You can see there some sequence, some DNA sequence. And this sequence, you can see the coding RNA sequence. And the gene, what is the function of the gene? This is the first chapter we studied. The second chapter, in our human being's body, all of the genes forms the genome. What is the genome? What is the genome? All of the genes in our cells, it is a genome. And what is the function? It's on. We study the first, second chapter. The third chapter, we begin to study the replication. Maybe some students, this part we already studied in the middle school. You can see through the originations and the elongation and termination. You can see maybe finished the replication. But in fact, this part we furthermore we studied for the different organisms. You can see different location orientated the replication. Here we give names orientated point. And how about the process of the replication? We furthermore we studied. And second, the fourth chapter, we studied the DNA damage and the DNA repair. Normally, you can say our bodies, our cells, our DNA meet all kinds of factors. The environmental factors, the all kinds of substances you can see can result in the DNA damage. For example, the ultraviolet light, you can see strong energy. You can see can make the DNA. You can see cleavage, maybe the mutation. At that times, you can see all kinds of the physical factor, the chemical factors, the biological factor make the DNA. You can see maybe change the structure, maybe destroy the structure, and at that times forms the DNA damage. In fact, in our body, you can see the DNA damage must and maybe immediately repaired. At that times, you can see how to repair this. How to repair? You can see the in our body. You can see several systems and prepared to repair the DNA damage. This is the fourth chapter we started, and the fifth chapter we began to study the transpiration. Maybe we studied. I teach this. Maybe students think this is very easy. Also, we already studied. In fact, the thoroughly progress, the thoroughly every step, maybe all of the factors bonded to the DNA, what kind of the space, how to begin choose the produce the RNA, the complementary RNA sequence. So this part we studied, and another one, all kinds of the RNA you can see may be produced. This is the original RNA. This kind of RNA you can say immediately need to be modified. Maybe need to the editing. Maybe need to the splicing. It is so on. So at the times, the sixth chapter, we studied the poster modification and the editing. It is so on for the originated RNA. And another one, the seventh chapter, we begin to study the, you can see, translation. Just you can see transcription from the DNA produced the RNA, the mRNA coding the amino acid sequence. This we studied the protein. We already lose, you can see, the mRNA sequence coding the amino acid sequence. So the Stevens chapter, we studied the translation and produced the paper tight chain. All kinds of paper tight chain produced, and at that times, this kind of the paper tight chain always low activity, low function. So at that times, you can say all kinds of the protein, all kinds of the paper tight chain synthesized in the ribosome. And at that times, from the ribosome, you can see synthesize and release the paper tight chain. And at that times, the paper tight chain need to be modified, need to be transportation. So the 8th chapter we studied, you can see the DNA of the protein synthesize, and at that times you can see maturations, modifications, and transportation.
Are you
What's
still
wrong?
on the first slide?
Yes.
Okay. Okay.
And totally, I introduce what we need to study in this semester, in this subject. Do you understand?
Yes, Lauscher.
So this part you can see synthesized in the ribosome. And furthermore, you can see the protein transported in the organelle, all kinds of the organelle, mitochondria, lysosomes, and transported to the ER, and transported to the nucleus. It is so on. In the cytoplasm, synthesize the peptide chain, and later you can transport it to the different location, different organelle. Other the protein you can say anchor, transported into the membrane and anchored on the membrane. So you can see the 8th chapter we studied. You can see the protein synthesized and modified and transportation. And the last chapter, the 9th chapter, I want to teach all of you the single transduction. You can see the single, the stimulator. How to transport the single to the steel. So at the beginning, I totally introduce all of you what need all of us to study in this semester in molecular biology. Okay. And also we begin to study the first chapter. You can see here all kinds of the picture, the beautiful. You can see zebra, and here you can see red blood cell. Here you can see the flowers, the goose, the babies. It is all kinds of the organisms. You can see different place, different site, presented the different color, different organization. What designed this? What designed this? You can see the zebra. You can see one line. You can see the white, the blank, white, blank, like this. What designed this? This. And to north windows, you can see it is the gene. The genes designed the property, all kinds of the structures, the properties designed by the gene. Okay. And first, you can see we recognize all kinds of organisms, all of the structure. You can see, and the beginning, we can divide the sticks, the on sticks, the long hairs, the blank eyes, it is so on. You can see with the development of the all kinds of the technology and the development the equipment. All of this, you can see the human beings, you can see recognize the organisms from the overrules level to the steels level. Leeuwenhoek, you can see, find their micro scope. So at that time, so you can see can be look clearly at the one steel. And furthermore, and also you can see use all kinds of the sequence technology. We can notice all of the molecular level about the all of the molecular the property. In our book, mainly focused on the nucleic acid molecule and the protein molecule. So here you can see molecular biology. It is the new subject that studies the life phenomena and their mechanisms on the molecular level. Nucleic acid molecules, the proteins, the molecules, it is on. This subject and knows it is the frontiers area where the life science developed fast and is widely intersecting and the permitted with other subjects. For example, maybe crossed with the pharmacy and forms molecular pharmacy. On the molecular level, studies the laws and mechanisms of the pharmacy. So this subject, you can see all kinds of the technology used in the other stable kind of the property. Oh, it's a stable subject. The pharmacies, steel biologies, and this part you can see. Why like this? It is the all kinds of the biological technology founded. For example, and before the sequence technology founded, and all of us don't know about the our genome composition. And knows is okay. You can see all the several chromosomes, all of the nucleic acid molecule, you can see bonded with the histone and forms the chromosome. All of the chromosome carry the genes. Here you can see. This picture last semester we studied. All those, the crick and the medium, founded the DNA double helix structure model. In fact, another infamous person, the Franklin. This you can see. Doing scientific research, you can see here the right site, you can see here, X-ray diffraction of the DNA. Finished in 1952. And how to form this kind of the structure. And later, the Crick and Meeting decoded this. You can see Worsing and the Crick. You can see forms this kind of structure. Okay. And these two persons, you can see Aquarius Nobel Prize in the medicine and the physiology in 1962. That's you can see molecular medicine. You can see when the molecular biology penetrated into the every branch of the biology and the medicine, it has promoted the development of the life science and the medicine in all facets, such as the pathogenesis research, diagnosis, and the treatment of the disease. At the same time, and promote the medicine development also is faster. And to north, you can see maybe and north you can see in China every days the people you can see doing the nucleic acid check and to find how about the person if maybe carries COVID-19. So this is the classical case. You can say use the molecular biological technology used to choose the medicine. Yeah. Another things you can see. Maybe some people like make my eyes more big. I feel is more beautiful. So and the times you can see. Watch. I just explain for you. You can see the property designed by the spatial gene. So and the times you can say maybe in the future I may be editing my gene and maybe make the property some property may be changed. And now you can see, for example, you can see use is very useful under recent several years. You can see the CRISPR-Cas9 technology. You can say easily maybe destroyed the gene. For example, here you can see this figure, the left figure. You can see the genome. This is the larger nucleic acid molecule in one cell. At the times, you can see, depending on the spatial sequence RNA, and made it the CRISPR enzymes recognize the spatial sequence and cut, destroy, and some sequences are destroyed at the times you can see maybe destroyed the function of this target gene. So, and later you can see here, target genes knock out. So, and the times, Bigelow J's DNA sequence was linked to the vector in vitro and directly used to mutagenesis spatial gene in one gene. Maybe some students have the interesting for this new technique. After class, you can search some more information. can be search some more contents studied. Maybe in the future, I planted the wheat, I planted the rice, and so on. Maybe I want to make the rice, the white, more higher production. And a lot of times maybe use this kind of the technique, change the genome structure. Maybe decrease the expression of the no useful gene. And here you can say making things of the molecular biology. You can see many studied the structure and the function of the biological macromolecule, especially for the nucleic acid and the protein. Yeah. Here, at the beginning of this class, I introduce all of you this semester what need all of you to study there. Focus on the structure and the function of the nucleic acid and the protein. And the interaction between the biological macromolecular and its relationship with the disease and the development of the organism. For example, this key case. In this case, you studied the Griffiths experiment of the transformation. You can see here. You can see different microorganism inject to the mouse body, and that times you can see. For example, you can see here marked with the red color and enclosed with the polysaccharide coat. This kind of the microorganisms is soft and count that count pentatonic strings. And second, you can see living pentatonic strings of the ace pneumonia. And this you can see the mouse. You can see maybe quickly and effect with this kind of microorganism, and the mouse died. And another words you can see, and the living injected the mouse with the living non-pathogenesis strings of the ace pneumonia. This mouse you can see, because this kind of the microorganisms without the pathogenesis property. So, and that times you can see the mouse living normally. And the last lines you can see, we inject the mixture of the hedging killed the pathogenic and the living non-pathogenic strains of the ace pneumonias. At that time, mouse died. And furthermore, and check and find the died mouse in this mouse body and find the pathogenic streams of the ace pneumonia. What about this experiment. You can see we think the organisms you can see affect the mouse lead the normal coat structure, neither the polysaccharide coat structure. And the normal low pathogenic strains never killed the mouse because this kind of strains will alter the pathogenesis property. So at last you can see it injected the mixture, and that times the heat killed the pathogenic strains of the microorganisms. Can be depend on the second kind of the own pathogenic strains coat structure, and maybe affect the mouse. And in the future, depending on this kind of organisms in the future, you can see it can be synthesize the DNA and infect the mouse cell, and later you can see makes the mouse die. And furthermore, detect the living pathogenic strains of the S. pneumonia. So at that time, we easily understand, you can see. When hitting killed this pathogenic strain, at that time, the DNA keep normally, never destroyed. So at that time, mixed with the non-pathogenic strains, and furthermore, maybe can be founded the offspring. Another one you can see the hatchery and the cheese you can see did the use the virus doing the scientific research and find the genetic material. It is the DNA. And at that time, so you can see the marked the DNA with the this kind of the for 32 acetous elements, phosphorus elements. At that times you can see, and affected the bacteria, and later cultured, and later also easily detect the 32 marked the phosphorus iso elements labeled DNA in the proteinase phase, but at the same time, and marked the another kind of the elements, sulfurs. At that time, so you can see, because this kind of elements you can see presented in the protein, no nucleic acid. So at that times, marked the virus, marked the phage, the sulfurs, and later they were detected this kind of the virus. So at that times you can see the directly elements result and certificated the genetic material. It is the DNA, no protein. So here, this is the directed experiment, the Hershey's and the cheese elements certificated the nucleic acid. It is a genetic material. This you studied in uh, so another one. This I simply explain something. Uh, you can see, and the experiment you can say certificated the DNA. It is semi-conservative replication. You can see at first the organisms you can see forever consider the in the meeting. The organisms you can see at the times you can see through the many stages. At the times all of the DNA molecule you can see the nitrogen marked with the 14 nitrogen elements. At that time, this kind of the organisms you can see. Maybe you touched to the another kind of the medium. It is with the medium, all of the elements and the nitrogen elements. It is the 15. Another iso elements. So at that times you can say cut the one times. At that times you can see forms this kind of the hybrid DNA molecule. Maybe furthermore, culture in this kind of the mediate at that time maybe forms the oh here at the beginning the organisms cultured in the 15th nitrogen iso elements mediate. All of the DNA molecule you can say, and first it is the nitrogen 15 contains this kind of the DNA. And one times cultured in the mediate with the 14 nitrogen mediate. At that time, so you can say forms this kind of the hybrid DNA molecule. And furthermore, you can say cultured in the 14 nitrogen elements mediate. At that time, so you can say the hybrid this fat you can see later change. And here you can see forms the light fat. You can see increase. So and directly replicated certificated the DNA replication. It is semi-conservative. Another one you can see here, and one Japanese molecular biology scientists and doing many research and find you can see the DNA sequence you can see synthesize you can see by there is a semi-conservative replication. And another one semi constituted replication. You can see for the leading straight you can see one time synthesize. For the lagging straight you can see partially synthesize. And this I here totally refer to something, and later we studied thoroughly. And in 1958, the correction put forward the central dogma of the molecular biology. At that time, you can see what is the mean. You can see the DNA directed to replicate and synthesize the DNA. One. And second, according to the DNA, can be directed to synthesize, transported, and produce the RNA. And RNA, mRNA, you can say directed to synthesize the protein. So this figure, it is the first central dogma for the correction put forward. And later we lose, you can see, the RNA molecule, you can see, also can be reverse transcribed and produces the cDNA. And also like this. Another ones you can see the RNA molecule you can see can be act as the maybe you studied in the medical microbiology. You study you can see several kind of the RNA virus. You can see can be according to the RNA temperature and to synthesize the RNA. So, and north you can see is the central dogma and developed and north like this. DNA can be replicated and produces the offspring DNA. DNA directed to synthesize RNA transcription. The RNA directed to synthesize the DNA reverse transcription. And another one, according to the RNA, can be replication produced the offspring RNA. Another one, the RNA directed and translated to produce the protein. Okay, this totally introduce all those things we studied in this semester, and we reached 10 minutes. Reached 10 minutes, and we began to study the first chapter. At the north, I just refer to something about this semester we studied. And later, we began to study the important chapter, the chapter 1, the gene. Okay, reached 10 minutes.
Okay, Lausha.
Okay, Monsieur.
干啥了？下午不去打球了？哦，还有个请假是吧？运动不行，去打球就行。无聊。我的是今天晚上，我们学校的大做的实验，我搞那个啊，4点半吗？我喊着下回一起搞的这个我什么哪些搞第一，我去报项目的时间，他表是这样的，我不是我说的我把这个信息从人家上门打电话叫你报，我把信息发了，对不对？真是的这个。搞那鬼东西，他不是跟你去这块儿开个车去喝两杯酒，不是天有酒喝就明白。嗯嗯，干啥呢？啊，跑公司去鬼工了。下午吃两千吗？方便啊？手机你写的那东西我哪里我怎么下那个我还以为我打电话什么你直接发就行了，每次。嗯，我问哪个事情？就是不讲人了。嗯嗯，我放在写遗作，就是他那个东西不能是假的，然后你就直接通讯就行了。要是 C 的话，你一般你的研究方法啊，不讲可以，假如说工具条例是的，蛮影响的，是影响你多影响的。给你摇过一次试管，你是不是你这些？嗯， 那不是球拍那些都在厉害的吗？这样个下班有没有？你跟谁过去的？哦，那你要不方便，你就自己抓紧时间。你要方便，我就赶你。你说这边有点事，就过来问啊。朋友谈的怎么样了？我就比较是你做这个东西，我还是我一定来做事情况。这你感觉会影响我们俩的销售或者是还是这个也不是说强求。做这个东西还是说我跟你在一起，我还很有信心。我绝对不话不能放手，这个话不能就比。你只能跟你现在跟他交流的是，第一，我跟你感觉在一起的话，很好，是吧？对于我们的问题还是充满信心，从这个中心思想出发的。你不能是的，然后老人，呃，对啊，你说反正我是已经做好准备，做出100%的努力，是吧？嗯，不一定，这个你该是从这个角度给他说，你一点点也不是白。不能做成这么多。 好。Okay, come back.
OK，老师。
And today we begin to study the first chapter. You can see the genes. You can see gene. You can see one stretch of the continuous DNA sequence encoding the protein or RNA. Maybe some students think you can see the DNA derivative and coding. It is the RNA. And to know we know you can see the some sequence you can see produced the and directed to synthesize mRNA furthermore directed to synthesize protein. So all the times we see is it is encoding the protein. Thirdly, for the some sequence only encoding the RNA. For example, the tRNA, the Robertson RNA, the microRNA, the non-coding cycle RNA, low coding RNA, it is all. So one DNA sequence encoding the protein, one encoding the RNA. This kind of sequence we see is One sequence gives the names to one gene. Do you understand there? You can see the gene. In fact, it is the DNA sequence. This kind of DNA sequence has a function encoding the protein or encoding the. And chapter 2, we studied the genome. Genome, what is the genome? Genome, you can see it is very easy. All DNA sequence in one cell, including the gene, including the non-coding sequence, maybe act as the structured DNA sequence. So all of the DNA in one cell, we say it is the genome. Totally. Here, I emphasize for my students, except the nuclear DNA sequence, my students don't forget, for human being, mitochondria, this kind of organelle, also contains the DNA. So for human beings, still you can see the nuclear DNA sequence, it is the mitochondrial DNA. For here, you can see this table. This is for this English words, the gene. The first times you can see ginseng. The first used the words gene. In fact, the genetics, the Morgan founded the James theory. You can see the code. At that time, this the Morgan related the James to the property. And later, you can see the bundles and the totem founded the one James, one is done. Partially right, I think here we studied, you can see one enzymes, maybe several peptide chains may be formed. So one genes, one enzyme, partially right. And the 1950s, you can see Benzer founded the system, doing scientific research and founded, used the virus and founded the system. And the 1960s, discovered the genetic coder. You can see all kinds of the amino acid. You can see according to the mRNA sequence, you can see can be discovered the peptide chain. From here, you can see is the mRNA to the peptide chain is easily founded. And 1990s, you can see forms the modern conception of the gene. For this, you can see this you study the genetics. You know, you can see the Mendel doing some famous experiment and studied and presented the law of the segregation as the lowest independent statement. At that time, what decides the property? You can see the Morgan or Mendel hypothesis, the genetic factor. Genetic factor decides the property. This determines the heredity. What is the genetic factor? Gene. And the second, you can see the ginseng. And this person doing scientific research and find the emitter in his book introduced the terms the gene. At that time, what kind of substance it is the gene? Don't know. Don't know. And this person, and furthermore, presented the maybe some genetic material determines the property. This genetic factor composition together, it is the genotype. And another, you can see maybe presented the higher the long or the higher height, maybe it's right, the redder colors, the green colors. It is one. It is the phenotype. Okay. Another person, you can see the Morgan. Hey, and doing some research and the fruit fly. Use this kind of the animals doing scientific research and suggested the gene was mapped on the chromosome. And at the times you can see directly related the gene to the certain substance. And furthermore, the genes is viewed as the fundamental unit of the mutation, changing, and the function. And to long we notice you can say one base may be changed forms the mutation. Maybe form changes the structures, changes the function, and so on. This two person. This two person, it is the biochemist. You can see the one genes contains the information for the produced the spatial enzyme. One genes, the one enzymes. Partially right. Okay. And furthermore, you can see in 1951, the Yalofsky presented find one genes, one paper on the gene. Also, it is the partially right. And later, doing scientific research and find the produced among you can see can be forms different splicing, maybe forms the different produced different kind of the peptide chain. Maybe one genes, one sequence, you can say maybe produced the several peptide chain. So also you can see here as this, it is the partially right. Bender doing some research and use the various and presented the zero of the statistron. What is the statistron? You can say one unit of the genetic function defined by the complementary test, which corresponded to the one open reading frame, ORF. Means you can say one genes, it is one statistron. Okay. Just video. I simply introduce for all of you the history of the gene. You can see genes are the basic unit for the storage and transmission of the genetic information in the cell and the individual. Deciding the genetic traits and be found in the chromosome and the mitochondrial DNA. So this is thoroughly, you can say, answer. You can say what is the genes, the function, and the locations, and so on. You can say the genes encoding all nucleotide sequence of the RNA and the protein sequence, and also includes the related regulations sequence. So let we begin to study the structure of the gene. You can see one gene includes the coding sequence. The coding sequence gives the names. It is a structural gene and the added regulations sequence. And DNA used to form the genes in most organisms, and few of the organisms, the substance, the genetic material, and forms the gene. It is the RNA. Okay, the structure. This part I studied the quickly. Primary structure. This if you forget something and need all you revise reviewed this part in biochemistry. DNA primary structure. It is the nucleotide sequence. Linked 3 prime 5 primes phosphodiester bond. Backbone, you can say phosphate, pentose. Direction, you can say 5 prime to the 3 prime.